Golgi is referred to as the cis face, and the pancake that's on this side of the stack, closest to the plasma membrane, which is shown right here, is referred to as the trans face. Proteins will move through the stacks of the Golgi and obtain different modifications as they move through the stacks, or the cisternae. And they move from the cis face of the Golgi to the trans face. Now let's look at organelles that are involved in breaking down things in the cell. First, let's look at the lysosome. The lysosome is the recycling center in the cell. It contains enzymes called acid hydrolases that break down the major biological macromolecules into their monomers, and then those monomers can be reused by the cell. So, for example, it breaks down nucleic acids into nucleotides, breaks down proteins into individual amino acids. They're called acid hydrolases because they function in the acidic environment of the lysosomes. The lysosomes have a higher concentration of protons compared to the cytosol. In fact, the pH of the lysosome is only 5, but those acid hydrolases function really well in that low pH or high acidity environment. If they were ever released into the cytosol, where the pH is about 7.2, they wouldn't function properly, and all of your macromolecules would remain intact. The second organelle that breaks things down in the cell is called the peroxisome, shown here. Notice that both of these organelles look similar. They're both spherical organelles that are enclosed by a single membrane, but they have different functions. The peroxisomes are involved in redox reactions, and we'll talk a lot more about those in class. Okay. And some of those redox reactions that are involved in breaking down molecules generate peroxide. Okay. And that's why this organelle is referred to as the peroxisome. Now, peroxide can be very toxic to cells. It damages molecules, but fortunately peroxisomes also have enzymes that break down and get rid of the peroxisome, get rid of the peroxide. Another important thing that's broken down in peroxisomes are fatty acids. And some things that we don't want are also broken down in peroxisomes. For example, in your liver, peroxisomes break down the ethanol that is part of alcoholic beverages that you might enjoy occasionally. Okay. Next, we'll take a visit to my favorite organelle, the mitochondrion, or mitochondria is plural. The mitochondria is often referred to as the powerhouse of the cell because this is where the majority of ATP is produced in the process of oxidative phosphorylation. So it produces ATP. I'm going to abbreviate it as oxidative as OXPHOS for oxidative phosphorylation. Now, the mitochondrion is the second organelle that's enclosed by two membranes rather than one. Let me show you in a little bit more detail down here. So there's an outer membrane, and then there's an inner membrane, also called the cristae, which has lots of surface area. It's highly invaginated, and that's where the enzymes that carry out oxidative phosphorylation are localized. Now let's look at some structures in the cell. These aren't organelles because they're not enclosed in a plasma membrane, but rather are composed of proteins. So the first are these blue lines shown here, representing the cytoskeleton. Just like our own bony skeleton, the cytoskeleton of a cell provides support. But another really cool function of the cytoskeleton is that it drives cell movement. So it moves things around in the cell. Sometimes, for example, the cell wants to move whoop, whoop, the mitochondrion around, whoop, maybe not outside the cell, inside the cell. May want to move an organelle around, may need to move a vesicle, a membrane-enclosed compartment around in the cell, and it does that using the cytoskeleton. But the cytoskeleton is also important in some cases for the whole cell to move. So, for example, your white blood cells moving to a site of infection involves the cytoskeleton. The other structure in an animal cell, shown here, is the centriole. Again, it's not an organelle because it's not enclosed in a membrane. It's composed of protein, and in fact, it's composed of proteins that also make up the cytoskeleton. It has this very distinctive two barrel-like structures in which the barrels are positioned 90 degrees to one another. It's involved in cell division. Specifically, it's involved in separating chromosomes during cell division. So we'll talk in a lot more detail about the structure and how the centriole works when we talk about the processes of mitosis and meiosis. Okay, now let's turn and look at some of the organelles and structures that are found in plant cells, but not animal cells. So here we have our generic plant cell. First thing I want you to notice is that you don't see centrioles in this plant cell, nor do you see lysosomes. Those are features that we only find in animal cells. Now let's look at what you find in plant cells, but not animal cells. First, you see a cell wall. Okay. The cell wall is important for structure and support. Okay. Notice that even though it has a cell wall, it also has a plasma membrane inside of that cell wall. Another feature we find in plant cells, but not animal cells, are chloroplasts. And this is where that very important process of photosynthesis occurs, in which carbon dioxide is fixed into sugars. So, sugars are synthesized in photosynthesis. So the chloroplast is the third organelle that's characterized by two membranes. Let me show you. I'm going to draw it up here. Okay. There's an outer membrane and an inner membrane. And the chloroplast also has structures inside called thylakoids. So these are membrane-enclosed compartments that are stacked up. Each of these is called a thylakoid, and the entire stack is referred to as a granum, or the plural is grana. Okay? And we'll look at more detail at the structure of the chloroplast and what's happening in each of these compartments when we discuss photosynthesis. The other organelle that you find in plants, but not animals, is a vacuole. And the vacuoles are a site of storage. Water is stored in vacuoles. So when you water your houseplants, you take care of them. That vacuole will increase in size as it fills up with water, and the membrane of the vacuole will push up against the plasma membrane in the cell wall, making your plant stand up nice and tall. And as you neglect your plant and it becomes dehydrated, the vacuole begins to shrink, and your plants, like mine, begin to wither. The other things that you might find stored in a vacuole are chemicals, like caffeine and also toxins that make plants toxic to some animals. And you also find pigments stored in vacuoles. So this is what makes your flowers nice and colorful. Okay, so that wraps up our tour of the eukaryotic cell. In class, we'll look in more detail at the structure and the function of specialized cells.